type of training that graduate students rarely get, and I learned how to talk about my science in a way that is engaging, enlightening, and informative. You can learn more about our innovative program at engage-science.com. And now to our second guest. Human bodies are made of trillions of cells. When these cells make healthy decisions, they can lead to diseases like diabetes and cancer. Our next speaker will explain how cells communicate information with their surroundings and the methods he uses to understand how these decisions are made. Please give a warm welcome to Sam Entwistle.
Almost there. All right, everybody, can everybody hear me? Yeah? Okay. So I research how to help diabetic cells make better decisions. But first, before talking about what that even means, I want to talk about ice cream. So I love ice cream. Whether it's in a cone or in a dish, ice cream is really a unique sensory experience. Visually, it spans a full color spectrum. And the smell of an ice cream parlor is really unmistakable, as are the memories that you might associate with it. But I think the main thing that makes ice cream so great is that it's sweet. And it's sweet because it has sugar in it. So part one. The sugar dilemma. So of course ice cream has sugar. But actually not all foods that have sugar in them are sweet. So carbs, for example, or carbohydrates. When we digest carbs, our body breaks them down until they become sugar. And so ultimately it has a very similar effect to the sugar that you would get from an ice cream cone. Now, we often think of sugar as something that's unhealthy or bad for us, but really, we can't live without it. And we need sugar because the cells in our body need sugar. Now, our cells are the smallest living unit of life in our body. And cells all over our body need sugar. Muscle cells, liver cells, fat cells, brain cells. These types of cells in particular, are some of the most hungry in our body. They need a lot of sugar. But how do these cells get sugar? So after we eat an ice cream cone, we chew it up, swallow it, it goes to our stomach. Our stomach dissolves the ice cream cone, and then it goes to our small intestine where it can be absorbed into our bloodstream. And from there, nutrients such as sugar can travel throughout our body to communities of cells all over the place. So the sugar, so most of the time we have sugar in our blood, pretty much all the time. However, after we eat a meal, the amount of sugar in our blood increases quite dramatically. But what happens after that? Well, the cells that live around our blood vessels, in particular the types of cells I mentioned earlier, they soak up sugar from the bloodstream. And they take the sugar into the cell and lower the amount of sugar in the bloodstream. Okay, so this is normally what happens after a meal. Now what happens to the sugar once it gets into the cell? So I told you that sugar is really important. And it's important because cells can use it for energy, and they can also use it, they can break the sugar down and convert it to raw materials that our body can use to build more cells or other structures. Now, I told you that sugar is a source of happiness. We all know that. And sugar is also important for our cells, and it's important for life. However, for many people in the world, sugar is also a major source of worry. So 1 in 12 people in the world today has diabetes. But actually, this disease has been around likely for millennia. Now, in the 17th century, diabetes was known as the pissing evil. And the reason for this is that urination, frequent urination, is a common symptom of diabetes. Also, the urine of people with diabetes often has a lot of sugar in it. And this is actually how diabetes can be diagnosed, which may be what's happening in this painting. But there's another reason why it was called the pissing evil. And that's because up until the 20th century, diabetes was basically a death sentence. So most of the problems associated with diabetes are because of high blood sugar. Now, when you have high blood sugar, there are some cells in our body that are just not built to handle so much sugar. And one example of these types of cells are the cells that line our blood vessels. And so when these cells take on too much sugar, they can be severely damaged. Damaged blood vessels, that underlies a lot of the really serious and tragic complications that happen to people with diabetes, such as blindness or kidney failure. So one major question that we face as people trying to understand and treat diabetes is how can we make or convince the right cells to absorb sugar? And the first answer to this question was found nearly 100 years ago. Part 2, insulin. So the story of insulin starts with dogs. And in particular, it starts with dogs that were part of this research study by Frederick Banting and Charles Best in Ontario. And Banting was a doctor and Best was his medical student. And they were interested in understanding the pancreas. And in particular, so the pancreas is an organ shared by both dogs have it and humans have it. And they had this idea that something in the pancreas could help and possibly cure people with diabetes. And to test this idea, they took pancreases from healthy dogs and they purified certain parts of their pancreas and then injected those purified parts into dogs who had diabetes. And amazingly, this cured those dogs of diabetes. And so future research turned out to show that the pancreas was actually releasing this substance called insulin. And insulin goes directly into the bloodstream. And so in these dogs who had diabetes, the insulin dramatically lowered their blood sugar and prevented these bad effects of high blood sugar on, for example, blood vessels. Shortly thereafter, pharmaceutical companies developed ways to generate large amounts of pure insulin. And this allowed people with diabetes to be treated. And many, many people's lives were saved because of this discovery. And this was enough for them to win the Nobel Prize. So Banting won the Nobel Prize along with another scientist who oversaw the study. Yet despite this, at this point, nobody really had much of an idea of how insulin actually worked. How is it actually lowering blood sugar? Since then, we've learned quite a bit. So insulin is the green triangle, and when insulin leaves the bloodstream, it finds its receptor. And Katie just talked about a few different types of receptors that sense opioids. There are many types of receptors, including ones that sense insulin. So after the insulin attaches to the receptor, it sends out a signal into the cell, a message. And the result of this message is that something called a transporter moves to the outside of the cell. And this transporter allows the sugar to enter the cell, thus removing it from the bloodstream. But this doesn't really tell the whole story, because what is the signal? How is the signal transmitted to these transporters? So to talk about this, I want to talk about high school. So there's one thing that you can find at, I think, any high school, and that is rumors. So this is the most popular couple in school. Say his name's Enrique, and her name is Sasha. And they're just like a perfect couple. Or they seem perfect, because one day they broke up. Oh, sorry. they break up. So we know that this news is going to spread across the school like a wildfire. But how is that actually going to happen? So the first thing that needs to happen is someone has to find out first. So in this case, let's say that Sasha's best friend Lucy finds out first because Sasha texts Lucy in the middle of class. But that, and Sasha also says to Lucy, do not tell it. But what Lucy does instead is immediately tell the three guys who are sitting next to her. And then one of those friends, he tells his girlfriend after class, and then she tells her two friends on the swim team. And then before the end of the day, everybody knows about this. Okay, so this way of transmitting news through like a chain reaction, this also happens in cells. Except instead of little high schoolers inside of cells, we have proteins. And I've actually already told you about a few proteins. Proteins are tiny little molecules inside cells, encoded by our DNA. And the receptor for insulin is a protein. The transporter for sugar, the blue transporter, is a protein. And insulin actually itself is a protein. But there are many other proteins in cells. Okay, so insulin finds the receptor. Oh, and then something happens to the receptor. There's these little chemical marks appear on it. And these are called phosphorylation. So phosphorylation, what is it? It's basically a way that proteins can communicate or gossip inside cells. Now, this protein on top gets phosphorylated twice, and when this happens, it almost turns the protein on, and it can then phosphorylate this purple protein at the bottom. Now, this is phosphorylation can be added to proteins, but it can also be taken off proteins, so it's reversible. And this is really, this idea of phosphorylation really transformed how we understand communication and the way information travels inside cells. And this actually won a Nobel Prize as well in 1992. And the two scientists who discovered this are two guys named Ed, two professors named Ed, Edwin Krebs, Edmund Fisher, and actually these professors both did their work at UW, which is cool. So phosphorylation, the insulin receptor gets phosphorylated. What happens next? The pink protein gets attracted to the phosphorylated receptor, it gets phosphorylated too. And that leads to another phosphorylation, and then more phosphorylation. And then, eventually, the transporter gets to the outside of the cell, sugar can get into the cell. Okay. So we have a more complete picture now. Insulin goes into the bloodstream, it finds cells that have its receptor, there's a wave of phosphorylation, and then sugar finds the cells and gets taken into the cells. So the power of insulin was able to cure many people of diabetes, and still does to this day. So diabetes is cured, right? So we all know that that's not remotely true. Actually, diabetes is really on the rise. Since 1980, the percentage of the population in the world with diabetes has almost doubled. So it turns out that insulin really only helps people with a certain type of diabetes. And in these people, they can't actually produce their own insulin. Their pancreas has, the cells that produce insulin die in the pancreas. And so taking insulin as a drug kind of is like getting like a replacement pancreas for that function. However, there's a whole other type of diabetes called type 2 diabetes. This is also called adult onset diabetes. Now this obesity is, people with obesity are much more likely to get type 2 diabetes. And because of this, the treatments for type 2 diabetes often are the same as the treatments you would do to just lose weight. So that means diet, and that means exercise. But there's a problem with this, and that's first of all, a diet and exercise regimen is just really difficult to maintain. But also, in people with type 2 diabetes, they're sort of already at a disadvantage because their cells are working against them. So what I mean by this is that if you give you give people with type 2 diabetes insulin, the cells that normally should respond to the insulin, they don't. You can give them even more insulin, and still nothing happens. And so now the new question is, how do you convince these cells to respond to insulin, or how do you get them to behave in a better way? So it leads to part 3, helping cells make better decisions. So I'm going to go back to ice cream. So decisions are hard, right? That includes decisions of small things like what type of ice cream to eat. And there are a lot of different things that can influence decisions. So there are things from like the outside world. So for example, advertisement. You see an advertisement for Ben & Jerry's online, or like see one in a magazine, then it sort of plants a seed in your mind that like, that'd be like pretty good to have some cherry rice seeds. Or perhaps a friend has a cone right there next to you, and you kind of want one as well. So those are like outside influences that can determine like what type of ice cream you want to get. But there are also internal influences. Now, an example of this could be if you're allergic to peanuts, you wouldn't choose an ice cream flavor that has peanuts in it. Or if you just sort of have a particular flavor that you just know is really good. So I'll give a shout out to cookies and cream. So cells actually have external influences and internal influences as well. And insulin is an example of an external influence. So that's something that comes from the pancreas, or it comes from like an insulin therapy, and that's a signal from elsewhere that tells the cell to do something, in this case, to soak up sugar. But there are many other things going around in the bloodstream as well, not just insulin. And some of these might have important messages as well. But that's not even all. Our brain is sending tons of messages to our body all the time through electrical signals. And these can often be really important and are something that the cell probably wants to pay attention to as well. And the neighboring cells around the cell, they can kind of sense the environment and send out little chemical signals that kind of report on what conditions are like. And those are probably important to listen to too. So there's sort of a lot of things that the cell has to do. Okay, so those are external influences. So what about things within? So let's say a cell, there's too much fat buildup inside a cell. So that could be kind of something that the cell should pay attention to and respond accordingly, as well as maybe an overload of sugar, or an overload of some other type of nutrient. Or protein damage. If proteins get defective, this is something that the cell needs to recognize and do something about. So these can all kind of distract the cell and influence its behavior. Okay, so we know that cells, it's good, or we want them to be able to sense and respond to insulin. But there are so many other things as well that they need to be able to pay attention to. Okay, so how do cells manage this? It seems like you would almost need a brain to be able to do this. So do cells have brains? No, they don't, but they have proteins. And so the way that proteins cooperate and interact, and yes, gossip, helps to determine the decisions that cells make. So proteins within our cells, through phosphorylation, are sending little bits of news to each other, and this is happening like all the time in cells, throughout the entire cell. It's almost like the cell is a city in itself, with like all these little messages being sent between these proteins. So how do we make sense of all this? So in order to do this, this is what I study. So in order to do this, I can measure thousands of these phosphorylation events in cells at the same time, when cells are kind of placed in different environments, and see if I can make any sense of how these phosphorylations are determining the decisions that cells make. Now this is not easy. Now, so as an example, I told you about insulin leading to some phosphorylation changes, and it leads to actually a whole lot of phosphorylations, excited proteins phosphorylating each other. And this is all like sort of part of a comprehensive response to insulin. But what if these proteins can get distracted? Let's say there's a signal coming from elsewhere in the body, from the brain. Or let's say there's something coming from inside the cell. All of these things could maybe distract the proteins in the cell from responding in the way that they should, that would benefit the health of the person the most. Now, ideally, we'd like to have some type of new therapy, like a drug, that helps to refocus the attention of these proteins and to respond to these really important signals, such as insulin. And people with type 2 diabetes, they really desperately need a therapy that's reliable, that lowers the amount of sugar in our blood. A therapy such as this could potentially cure type 2 diabetes. So how far are we away from this? So in order to gain a real understanding of how phosphorylation affects the way cells make decisions, we still need to learn basically like what the language of phosphorylation is, and how cells communicate, how the language of phosphorylation, and how cells communicate little bits of news. And we need to understand how these all sorts of different sources of gossip inside the cell can be processed together to come to an overall decision by the cell. But the more we learn, the closer we're getting to bringing better therapies to people with type 2 diabetes. And this is good because summer's coming, and everybody deserves to fully enjoy this one. That's all. So I'd like to acknowledge the Engage program. This has been a really awesome experience, and Seattle Town Hall, and my graduate departments at the University of Washington, and then all of you for being a great audience. I'd be happy to take any questions. Yes.
I guess I kind of have a question. So I was going to ask, more specifically, what phosphorylation is, and what you just said, because we don't know.
So we do know what it is. It's a specific molecule made of phosphorus and oxygen that gets attached to proteins. It gets kind of chemically attached to proteins. And why that's significant is that by doing that, it can actually change the shape of the protein just a little bit sometimes, but it can change the shape enough that the function of that protein and the way it behaves also changes.
So when you said you're like phosphorylation going on with the proteins, is what you're able to observe that molecule? Does it move are they sending molecules?
No, they're not. So the way the sending happens is you have like one protein might get phosphorylated and its behavior changes, and if that protein itself can add phosphorylations to other proteins. So let's say the phosphorylation basically acts as a switch that turns on that protein and allows it to then add a phosphorylation to another protein. And then that flips a switch that allows that protein to then go on and add phosphorylation to another protein. That's the idea.
Where does that molecule made of phosphorus and oxygen come from? Is that just kind of floating around in the cell and gets attached to the protein, or does the protein somehow get it from somewhere? And how do more of them come when it passes a phosphorylation between proteins?
That's a good question. So the phosphate is what that whole thing is called, the phosphorus and oxygens. That comes from a molecule in the cell called ATP. And some of you are nodding your head. ATP is like a major. It's kind of like a source of energy in the cell, and that's one of the main things. So actually, when I said that sugar makes energy inside the cell, that's like one of the main things that it makes. And that's sort of like an energy currency in cells. And it can also be used to add phosphorylation. questions?
What sort of equipment do you use to watch the
Yeah, that's a good question. So I could talk for ages about this. So I use something called a mass spectrometer. Mass spectrometers, they're in general really useful for studying proteins. But they, basically what we do is we can like take all the proteins in a cell and chop them up into little bits and then measure those little bits in the mass spectrometer. And some of those little bits have the phosphorylation still on it, and so we can measure those little bits with the phosphorylation, and that tells us how much of that is coming, exists in the cell.
That's a great question. That's fun. So what type of cell do you use, and where does the cell come from?
So I study a type of cell called brown fat. And I actually didn't talk about brown fat at all this talk. But brown fat is, it's a tissue that we have it in humans, but it's still like a little bit, we have like a very small amount of it. And basically, brown fat is really, really good at soaking up glucose from the blood, sugar from the blood. And I'm trying to understand kind of how it makes its decisions to see if we can get it to work in like hyperdrive. And if we can do that, then the brown fat's really like soaking up a lot of sugar from the bloodstream, and that might be a really fruitful direction for type 2 diabetes.
Do you know why people develop type 2 diabetes? Is that also
So in general, we know like risk factors. We know like obesity is a risk factor, but like genetics, depending on your genetic background, that could be a risk factor. And then like socioeconomic factors as well. But like, are you asking, what level are you asking? Like kind of at the cellular level, or more broadly like the how do people get?
I don't know.
Okay. Yeah. So that's like it's largely a mystery. We know things that contribute to people getting it, but it's hard. It's something that's hard to predict. And then, right, exactly. Anything else?